What are the key differences between WES and whole genome sequencing? Imagine trying to read a book by only looking at a few pages versus reading every word from cover to cover. That's similar to how genetic testing methods work. Today, we're exploring two popular ways to analyze DNA: whole exome sequencing and whole genome sequencing. Both are powerful tools, but they serve different purposes and have unique features. Whole exome sequencing, often called WES, focuses on just the parts of your DNA that actually make proteins. These are known as exons, and they make up about 1 to 2% of your entire genome. Think of it like reading only the chapters that contain the main story. Because it targets these specific regions, WES is faster and costs less, usually around $1,000 to $2,000, and results can come in about 6 to 8 weeks. It's especially useful for diagnosing hereditary diseases because most disease-causing mutations happen in these protein-coding areas. On the other hand, whole genome sequencing, or WGS, looks at every single base pair in your DNA. This includes not just the exons, but also the non-coding regions, parts of the DNA that don't code for proteins but can influence how genes work. WGS covers all 3 billion base pairs of your DNA, giving a complete picture of your genetic makeup. Because of this, it's more comprehensive but also more expensive. Costs can range from around $3,000 to $5,000, and analysis takes longer, often up to 12 weeks. WGS can detect genetic variants in areas that WES misses, like regulatory regions or mitochondrial DNA. This makes it valuable for complex cases or research, especially when doctors suspect issues outside of the protein-coding parts. WES, however, is more targeted and efficient for finding common mutations linked to hereditary conditions, making it a popular choice in clinics. Another key difference is data volume. WGS produces a huge amount of information, requiring advanced computers and storage. WES generates a smaller dataset, which makes it easier and quicker to analyze. Also, WGS can identify structural changes in DNA and variants in mitochondrial DNA, which WES usually doesn't cover. In summary, WES is a focused, cost-effective way to find mutations in the parts of DNA that directly affect proteins. WGS offers a full view of your entire genome, capturing more types of genetic variations but at a higher cost and complexity. The choice depends on what your healthcare provider needs to know, your budget, and how detailed a picture you want of your DNA.